giving us a total capacity of about 5 terabases a month. The lab's been used for some complex, large-scale studies. In fact, since its inception, it's run about 2,000 whole genome and tumor samples, about 1,000 array-seq and exome-seq runs, and lots of smaller things, microarrays, real-time PCRs, etc. So we actually started out with the smaller machines, the GA2X. They generate a lot less data than the Illumina high-sec machines. So what actually happened is when we got the 3 high-sec 2000s, we were flooded with data. The shift was quite a challenge. The most obvious challenges came from sustaining a high throughput, a processing throughput that could match the data generation rate. The other challenge came from storage capacity, finding enough space to put all this stuff. But there were also many logistic and data management challenges, such as increased operator efforts and the increased number of samples that we were running. Tracking the provenance of the data, so remembering or somehow documenting how you generated some result from all those data sets that you generated. Documenting your analysis procedures so that you vary a parameter, you remember. Finding files, generate thousands of files, and even finding them is a challenge. So we devised a solution to try to get a grip on the situation and help us scale. Part of the solution is hardware. So we have a computing cluster of about 400 nodes for a total of about 3,200 cores. Each node is equipped with 16 gigs of RAM and a single hard drive for 250 gigabytes. We have additional machines to run services and run virtual machines. The network, each node, or rather nodes are grouped in 16 node enclosures. Each one runs a 10 gigabit Ethernet connection to the backbone, which runs at 40 gigabits per second. Our storage infrastructure is a 4.3 petabyte parallel shared storage system. It runs GPFS. A note about all this stuff is that it's actually shared. So it's not dedicated to our sequencing operation. It's shared within the whole research center. And it's also housed in our own computer room. So, as I said in the beginning, there was panic. Analysis was being done with the standard Illumina tools and scripting. Samples were being tracked in Excel, and the emergency was the norm. So we created a process with these features to try to help us get through everything. So we monitor the data acquisition devices, and when they finish running automatically, we launch the primary processing procedures. We record provenance information for all the data sets that we generate, and we have a system that is designed to achieve, or sustain rather, a processing throughput that is superior than our peak sequencing capacity. The system is devised with these 6 components, and I'll go through them one at a time. So sample submission, this is let's say the interface of the laboratory or system. People in the lab, so we use Galaxy in a modified way. We customize the system and add some features. People in the lab can actually submit the samples through the interface that we have here. They can register the samples that they're going to be sequencing, the flow cell on which going to be placed, and if it's a multiplex runs, they can associate barcodes to them. Okay? So going from Excel to this kind of system allowed us to move error checking, for instance, and all sorts of automated validation to the time where the information is being submitted. The system has a web interface, or rather a web service API, so they can be queried from the other components of our infrastructure to extract information about the samples and the flow cells that are coming through. And I guess the difference with the original Galaxy system is that in addition to the submission, the sample submission system, which was modified slightly and extended, we've added features to help or to let the people in the lab track the processing of their samples. So they can see when processing is started, how it's going, if there's an error, if there's a problem, it'll be recorded here, and then it's done. The next component is iRog. Some of you have used it, some of you have heard of it, and some of you maybe not. It's a file catalog, so it's a place where you can register all your data objects. It lets you, it's a layer of indirection between your storage infrastructure and your users. So what this allowed us to do is, as we were growing and we got new storage equipment, we ended up having to either transfer data from one storage to another, or dismiss some old storage infrastructure. Through iRods, basically this became transparent. So the users didn't have to know that their data was actually being moved from one place to another. It helps us track files and find them when we need them. We use the system to tag data sets with sample IDs, with the FOSO ID from which they come through. And NRODS has a pretty complete query system through which you can actually query these tags and find the data that you're looking for relatively easily. Also, sometimes we have geographically dispersed collaborations. So we need people to have access remotely to their data or to the data that we're generating. And through iRods, we can help them do this easily. You have a question?
You prepare questions afterwards? Or?
I beg your pardon?
You prepare questions afterwards or in between?
I didn't understand.
I mean, you accept questions afterwards or can I ask right now?
Maybe later.
Okay.
Okay, thank you. Okay, Omero Biobank is the next component in our system. This is a system that we've developed at CRS4. What we use it for is to track all the operations that we're doing on the data. In essence, it's a graph database that is specialized for this kind of work, so biologically related work. The graph has two types of nodes. It has entities, which are data sets, and actions. And actions take entities as input and generate new entities as output. Associated to this node, we can include all sorts of metadata. Specifically for entities or data sets, we don't actually put the data in there. We just put a reference to, in our iRods collection, you can find the data set at this location. We might also include other metadata such as data format and sort of that kind of stuff. Operations, on the other hand, we actually take the workflow that we ran to generate that data set, serialize it, and stick it in that node. So that means that if we need to, we can go back and find the same input file. We can take out that serialized workflow, reinstantiate it, re-execute it, and regenerate the same output that was generated the first time, ensuring reproducibility of our analysis. This is an example of what the graph might look like. So if we read this backwards, these arrows point to the parent, so we can see that we have a VCF data set that was generated from a band to some action, and this band was generated from a FASTQ file, and so on and so forth, all the way back to the original raw sequencing output. The workflow manager, next component. The task of the workflow manager is to execute operations on the data and record what it does. The analyses, of course, they require multi-step procedures, right? So, and parameters for each one of these steps. So we represent each procedure as a workflow, we give it to the workflow manager, and we run it. In our system, we're using Galaxy as the workflow manager for a number of reasons, including the fact that to interface with the Omero Biobank system, Galaxy makes it easy for us to extract the operations that were just performed and record them in our action nodes in our Omero Biobank. Okay, compute engines. So, like I said, we had a flood of data when we got these sequencers. We had some bottlenecks in our process. We needed to find a way to improve our throughput. What we did is we implemented, re-implemented the bottleneck steps in our process as Hadoop-based application. So we're using CO, which is a Hadoop-based NGS data processing toolbox for a number of steps. We use it to go from the raw BCL files that the sequencer generates to convert those to QSEC files and to do multiplexing when necessary, align and sort reads with this toolkit. We're using Hadoop Galaxy, which is an adapter to let us run Hadoop-based programs from Galaxy. So that means that in our workflow, we can include conventional programs and Hadoop-based programs. And with this adapter, we can just run the workflow transparently. Also, the package includes some utilities to let us do compression of the files and file concatenation in parallel, so by distributing these tasks in our cluster. Special purpose tools. Sometimes it happens that we need to perform special analyses. We might need an ad hoc tool implemented for it. When throughput is a concern, usually we turn to Hadoop again, but rather than going directly to the Hadoop API, we usually go off Pireddu, which is our Python API for Hadoop, which is a little higher level than the bare Hadoop API, or SecPig, which gives us a scripting interface that is specialized for sequencing data to write some quick scripts and analyze your data using the Hadoop cluster. Like I mentioned, the cluster that we're using is shared, and to access it, we have a normal batch system. This doesn't play nice with the Hadoop paradigm, where instead Hadoop assumes that it has complete and permanent control of its nodes, right? So we had to figure out a way to make the Hadoop work with the batch queuing system. We developed this tool called Hadoopka for this. And basically our setup was a little bit like this. We have the Hadoop head node, the job tracker, which runs on a machine and is always on. And it's waiting for jobs. When we submit a job, we have another daemon, the Hadoopka daemon, that monitors the queues on this job tracker. And as the need requires, it allocates machines through the batch queuing system from the cluster and associates them to the Hadoop cluster. To do this, basically it just submits a program to the batch queuing system. When the program starts, it'll have a node all to itself. It'll fire up the Hadoop node, and then it'll monitor it. Once it sees a Hadoop node, that the Hadoop node has become idle for a number of minutes, then it'll just take it down and relinquish the machine and return it to the general population. To make this work, we also forego the Hadoop distributed file system. This is, in case you're not familiar, Hadoop comes with a component that lets you run work, distribute work across your cluster, but also a component that lets you store files, or store data rather, in your cluster. It chops up your data into pieces and spreads it around your cluster. The problem for us is that if we're going to relinquish these machines to the cluster, we should remove that data that was left there. Otherwise, it would be inaccessible as soon as the machines are taken over by other jobs. So instead, we just rely on our parallel shared file system, leave our data there, and we don't use the HDFS file system. The advantage is, well, the flexibility, especially because we can relinquish nodes without any issues, and also the storage capacity that we have available is not dependent on the number of nodes that we have allocated, because we're not using the local disks. Another notable of this is that HDFS has great scaling characteristics, meaning that as your cluster grows, HDFS gets faster with it. So it's always fast enough for your cluster, so to speak. For us, performance in these terms really wasn't an issue because, like I said earlier, our nodes are only equipped with a single disk, so they don't have great I/O performance. Given those characteristics and the fact that really we're not using any more than, say, 80 or 100 nodes at any one time, the parallel shared file system is actually fast enough to be comparable with the performance that we get out of an HDFS that was running on our machines. Okay, Automator, this is the last component. Basically, it's the glue that makes everything work together. It's a custom-made controller and middleware based around a distributed event dispatching system, meaning that we have a RabbitMQ instance with an event queue that is running. Components emit events and put them in this queue. The automator is listening on the other end, and when it gets an event, it runs some event handler that is made specifically for that action. In this event handler, it can do lots of high-level operations. It can move data, it can launch workflows, it can register data sets with those other components, so with the Omera Biobank, with IRONs, and so to speak, but it doesn't touch the data. It doesn't manipulate data, it doesn't generate new data sets. All that stuff is done through the workflow manager with Galaxy, so that we can document those things. This is an activity diagram of how an event would be handled. We have a run finished events, so this would be triggered when a sequencer finishes and we notice. That event goes into the queue, the automator picks it up and executes the appropriate handler for it, the run finish handler, which would register the data with IRODs, it would register the data sets with the Omera Biobank, and then it would emit a new event, which would in turn go into the queue and then fire another event handler. This is the turnaround time for flow cells. Basically, this is a histogram, number of hours that it took to finish everything, and this is, well, the number of flow cells. We can see that, say for problem situations, say things breaking, usually we manage within 24 hours that they have the data ready, the data through primary processing. When things go wrong, what happens? There's a number of places where we should improve. Many of the problems are caused by system-level issues, in the sense that, sure, it's easy to point fingers at the sysadmins where they're not around, but oftentimes when we have problems with workflows failing, it's because maybe there was a broken node, or rather a node broke while it was processing. The file system might have hiccups or misconfigurations happen. The point is that we should improve our software to make it more resilient to this kind of issue. A simple thing to do would be just rerun something or retry something that has failed. Also, the automator is implemented as a single-threaded program, meaning that it can result in cluster underutilization. If in our workflow we have a step that does not use the cluster, but rather has a monolithic step that runs on a single node, the entire automator will be blocked on that node until it finishes that operation. That's something that should be improved. Also, recovering from problems, excuse my voice, is still cumbersome. I have some water here. Excuse me. So, what I was saying that if we have a workflow that fails to finish, basically we haven't implemented the high-level tools to help the operators to restore the operation to what it was before the error. So instead, to save operator time, what we do is generally just delete everything and restart from scratch. A little wasteful if you wish, but it saves people time, and that's important too. So in conclusion, the system's been in production since July 2013. Compared to the way things were originally, we've reduced the number of people that are dedicated to running the primary stage pipelines from 4 to 1. To keep up with the lab's data production rate, we've had to do two things. One is automate as much as possible, and the other one is adopt scalable computing strategies. So in our case, Hadoop. To keep up with our own process, we've had to implement process tracking and adopt a data catalog to help us find it. If there's a takeaway message from this presentation, it should be that automation is crucial to scale the data processing operations. And that's it. Thank you.
Thank you, Luca. You didn't mention if your site is a distributed geographically, or it is a central
location. Sometimes we collaborate with people that are external, but still, it's just a matter of giving them access to the data that has been processed at our site. We also have a Galaxy instance that is accessible from the outside. So usually what we do is after primary processing, if there is such a collaboration going on, whatever data sets were generated, we'll make them accessible through that Galaxy instance that is accessible to the collaborators. So then from there, they can continue, maybe do interactive analysis or whatever they want to do directly on that external instance. Although everything still happens within CRS4. Then at some point, they'll have to download the results.
So you mentioned a problem about the automator, about the single-threaded. Did you try this Kafka, the new this is distributed event?
No, I haven't tried it.
Okay.
I haven't tried it.
But you mentioned also that you are using GFS.
GPFS.
IROs above the GFS, right?
Yes, yes.
So, but DDN actually shipped with the luster. Did you try the luster, or why did you ship with GFS?
We had a GPFS file system prior to this one, and I can't tell you why. I don't know the details, but we had a lot of problems with it, to the point that we actually even lost data at some point.
Probably you want to say a last file system before.
Luster file system.
Yeah.
Our last luster file system, that might be it. So I don't know if, who knows whether it was, I don't know whether it was a problem with luster, which is used throughout the world, so maybe not, or some combination of the hardware we're using and the file system.
If I can interject one phrase here, the problem was that we were using luster to serve the general cluster. Mm-hmm. So we had competition between jobs who generate lots of small files and jobs would generate large files. And luster in our case didn't work very well, at least the setup we had didn't work very well when the number of small files we were generating were large.
Okay.
And we ended up that when we bought the next large-scale data storage system, GPFS was more, adaptable to what we were looking for.
That makes sense. Yeah.
Okay, thank you. I mean, there's going to be time for discussions, and I'm sure you have a lot of questions to look after this. But so, thanks again, Luca.